Okay, in Chapter 6 here, we're going to be discussing what does it take to get the antigen presented to the B cell or the T cell. Okay, because they're not, especially the T cells, are not going to respond to some randomly floating by antigen. It has to be presented to them in such a way as that not only can they recognize it, but they will get the signals to activate themselves. Because remember, back Chapter 4, we discussed that with the MHC, you have to have a specific size, whether you're talking MHC class 1, class 2, specific size epitope in there, and that the T cell receptor will interact with the MHC, that you will get a induced fit and then conformational change, which will then allow the variable regions to interact with whatever epitope is present. So, how do you get the epitope loaded into the MHC? Because remember, our major histocompatibility complex. Two types, MHC1, MHC2, presenting to either CD4 T helper cells or CD8 cytotoxic T cells. Best way I have to remember 1 or 2, 4 or 8 is everything has to add up to 8. MHC class 1 times CD8 equals 8. MHC class 2 times CD4 equals 8. Best way to get to it. But before you can have something loaded into MHC class 1, class 2, you have to go through some antigen processing. Okay? Because remember, your immune system does not learn a virus. It does not learn a bacteria. What it learns is a part of the protein on the surface of a virus, or on the surface of a bacteria. So that whole protein has to be broken down to give you a specific antigen, and then that antigen has to be presented. Now, when you're looking at a generic cell, here we're looking at a generic cell, a cell that can take things up, a cell that can spit things out. Endosomal takes stuff up, fuse it to a lysosome, digest it down, or it can make stuff in the Golgi and then excrete it out using secretory vesicles. Okay, this is just a generic cell. But what you have is literally two different types of compartments. The endosome, which is actually the exterior environment entrapped in a bubble, which is why here in this figure they show the endosome has the same yellow as the space around the cell. That's to denote that it's whatever that is has been trapped in an endosome, along with a bacteria, a virus, or whatever. It's, the environmental contents. The cytosol in blue, well, that's a different environment. This is a more controlled environment. So you have these two compartments, and viruses or bacteria are going to get into either one using different takeaway message. Cytoplasm, cytosol, class 1, endosome or a vesicle, tap 1, tap 2, into the ER. Now, the number of these proteasomes and the enzymes that go through and ubiquitinate fluctuates. Normal low level there in all of our cells, so that our cells can go through and randomly, like I said, randomly ubiquitate things, proteins, send them through the proteasome, and then load through tap 1, so they can be loaded, tap 1, tap 2, so they can be loaded into MHC1, so that T cells can see anything that's in that cell is self and must be ignored. Interferon levels go up. That means that some CD4 cell or neighboring macrophage or something has been alerted that there is an active infection. So interferon levels go up. So do the proteasome numbers. So do the enzymes that go through and do the ubiquitination. Means now more random proteins are going to be going in, and some of those random proteins are going to be viral or cytoplasmic bacterial. Those will be cut up, loaded up, and sent in. Now we also see that there are what they refer to as drips. These are going to be defective ribosomal products. That's what the defective RI ribosomal P products. These are going to be improperly translated polypeptides. Maybe an intron was stuck in there. Maybe an intron, for whatever reason, got translated. Or the protein just will not fold correctly. Remember, there are chaperonin proteins that will come and help get that final tertiary quaternary structure for some proteins that are just having an issue. Well, if that doesn't work, well, then they'll be ubiquitated, sent to the proteasome, and then off they go.
The folding and assembly of MHC class I molecules takes place in the lumen of the endoplasmic reticulum. The initial folding of the class I heavy, or alpha chain, is aided by the chaperone calnexin. The partially folded chain is transferred to a second chaperone, calreticulin, which aids the further folding of the chain and the association of beta-2 microglobulin. Other proteins, ERP57 and tapesin, associate with the nascent class I molecule, which binds to the TAP transporter via tapesin to form a peptide-loading complex. The peptides that bind to the MHC class I molecule are generated by a large protein complex, the proteasome, which is found in the cytoplasm. The proteasome degrades proteins within the cytosol of the cell to produce short peptides, which are then transported through the endoplasmic reticulum membrane by the TAP transporter. In the ER, these peptides are modified by the action of an enzyme known as the endoplasmic reticulum aminopeptidase associated with antigen processing, or ERAP. ERAP trims the amino terminus of many peptides and allows peptides that are initially too long to bind to the MHC molecules. Some peptides do not bind to the MHC molecule at all. Others can bind, but are unstable. These are released from the MHC molecule, a process called peptide editing. Finally, a peptide binds to the MHC molecule with high affinity to make a stable complex. This causes the final step in the folding of the class I molecule to take place, and the dissociation of the peptide-loading complex. The peptide-loaded MHC class I molecule is now free to exit the endoplasmic reticulum and be transported, via the Golgi apparatus, to the cell surface, where it can be recognized by the antigen receptors of CD8 T cells.
So, as discussed in that video, we're going to be looking at what it takes to get a epitope loaded into MHC class I. Now, when you look at, we've got to remember, we're beginning here in the cytoplasm. We're going to be working from the cytoplasm into the ER. We're going to just assume Golgi steps, and then we're going to look at what it is produced and presented on the plasma membrane. Now, when you look at what's going on here, far left column, far left figure, we see that MHC class I, we've got the alpha chain primarily there. We have the beta-2 microglobulin off to the side. Well, the alpha chain is kind of stabilized by calnexin. Calnexin is a transmembrane protein that has a large component, as you see there. It holds the class I alpha chain there. As the beta-2 microglobulin comes in and starts inducing more folding into the alpha chain, it loses its relationship, its interaction with calnexin, which is then replaced with calreticulin. Calreticulin acts as a scaffold, along with ERP57, Tapicin to hold this misfolded class 1 MHC in place. When you're looking at everything here, you also see you have TAP there. TAP will be having a close affinity towards our Tapicin ERP57 calreticulin complex. So, in the cytoplasm, okay, the ER ready to go. We now have the cytoplasm. Ribosome makes quite a few proteins. Okay, it's constantly going. Gets done with one messenger RNA, goes to another, does its thing. Now, the majority, depending on who you read, going to be about 70%-ish of the polypeptides made by the ribosome will fold correctly, will be correct. The remaining 30-ish% is going to be what I called on the previous slide DRIPs. Okay, defected ribosomal products. Don't fold correctly, won't do. Somehow the RNA splicing didn't complete, wasn't completed, still has introns, whatever. It gets ubiquinated. That DRIP gets sent to the proteasome where it gets chopped up into peptide fragments. Now, here in the figure, the third panel from the left, it shows the peptide fragments just going to the TAP complex. Well, they don't just go there. Turns out there's a protein known as TRIC, T-R-I-C. It's a complex of proteins that act as a chaperone, and they will, well, as the chaperone does, they will carry these peptide fragments to the TAP, which then allows them into the lumen of the ER, where ERAP, the, the endosomal, endoplasmic reticulum, blah, blah, blah protein, goes through and cuts off the tails, or cuts off the carboxyl end of these polypeptides, leaving the amino terminus. Now, as the video showed, it's not just as soon as one comes in, it loads in, and that's the way it goes. No, it's going to be random. One comes in, loads in. Does it form a stable MHC class 1? No, gets bumped out. But next one, bumped, until one loads in to the cleft of the MHC class 1 that forms a stable complex. If it forms a stable complex, calreticulin, ERP57, Tapicin, as well as the TAP complex, that whole scaffold there disassociates. Because a stable MHC class 1 complex has a shape that calreticulin, ERP57, Tapicin cannot interact with. So when it hits that correct quaternary structure that has an epitope loaded into it, it no longer can interact with any of those structural scaffold proteins. Those leaving the area of the MHC class 1 that is now in a stable conformation is a signal to tell the cell to begin traffic that via vesicle to the Golgi. Goes through the Golgi, then cis-face, trans-face, out to the plasma membrane, where it can now be seen by a passing CD8 T cell. So, even though the class 1 may be complete, quote-unquote complete, in that all the parts are there, it's still not the right shape. Still not ready to go. Okay? It needs to have a peptide, that epitope bound into it, for it to have any activity. We see that the peptide loading complex, that scaffold of calreticulin, tapain, ERP57, they hold everything there in place as it's showing here. Okay? Down in here, you see this right there, the yellow right here, that is the MHC class 1. All the rest of it is scaffolding, holding it in almost the correct conformation for an epitope to load in. Now, as long as we eukaryotes have been around, we think there have been viruses along with us that have evolved right alongside of us, and they have evolved means of inhibiting the loading of MHC class 1 in the ER. You look at the first one there, herpes simplex virus 1, has a protein known as ICP47. Blocks the peptide that's going to be loaded into the endoplasmic reticulum. The peptide that TRK is chaperoning in, ICP47 will gum up the TAP works so that TRK cannot chaperone it in. Or you look at something, cytomegalovirus. Inhibits TAP. Inhibits the ATPs, the swapping of A triphosphate for the A diphosphate. So that TAP is now stuck in the off position. Other ones block tapicin function. Other ones will block transporters or ubiquitin, the ubiquitation activity. Stop the ubiquitination of a drip. Won't go to the proteasome, won't be cut up, then TRK can't chaperone it to TAP, can't be transported into endoplasmic reticulum. So we refer to these different viral protein as immunoevasins. They allowed the virus to evade certain steps of the immune